The structure and metabolism of glycogen. Glycogen is a major storage polysaccharide in animals like starch in plants. Glycogen mainly occur in liver and muscles, and the modest quantity is present in brain. Three-quarters of the total body glycogen occur in muscle. Glycogen is the branched polymer of alpha-D glucose. It consists of short chains of glucose residues, approximately 13 glucose residues, with ranges from 12 to 15. They are linked through alpha 1,4 glycosidic linkage in the straight chain with alpha 1,6 glycosidic linkage at branch point. The glycogen molecule is a sphere and approximately 21 nanometer in diameter. Its chains are either branched or unbranched, and they are arranged in 12 concentric layers, but here you can see 4 concentric layers. The branch chains each has 2 branches are found in the inner layers, and the unbranched chains occur in the outermost layer. The glycogenin, which is a 37 kilodalton protein, is a primer molecule for glycogen synthesis. Here you can see 1,4 glycosidic linkage and 1,6 glycosidic linkage. The glucose molecule in straight chains having alpha 1,4 glycosidic linkage and at branch point alpha 1,6 glycosidic linkage. Liver glycogen and muscle glycogen have different functions. Liver glycogen maintains the blood glucose concentration in fasting state, while muscle glycogen is responsible to provide glucose 6-phosphate, which enters in the glycolysis to provide the energy to the exercising muscles. As I have already told you, that liver glycogen maintains the blood glucose concentration in the fasting state. After a meal, the liver concentration of glycogen is about 450 millimole per liter glucose equivalent, and this level falls to about 200 millimole per liter after an overnight fast. The liver glycogen is almost totally depleted after 12 to 18 hours of fasting. Although muscle glycogen does not directly yield free glucose because muscles do not contain glucose 6-phosphatase, the pyruvate formed from the glucose released from the glycogen when it enters the glycolysis is transminated into alanine, which is exported from muscles to the liver, where it will convert into glucose through gluconeogenesis. The storage of carbohydrate in 70 kilo human being is as under. The total body content of liver glycogen is 90 gram, which is 5% of liver weight, and total body content of muscle glycogen is 245 gram, which is 0.7% of muscle mass, and extracellular glucose, which is 0.1%, is 10 gram is present in the body. Here you can see the structure of UDP glucose or uridine diphosphate glucose. This nitrogenous base uracil linked to the ribose through glycosidic linkage, and this nucleoside is called uridine, and to which this phosphates are attached, and so it is called uridine diphosphate. And when glucose is attached to it, then it is called uridine diphosphate glucose. Now we will study the pathway of glycogen biosynthesis from the glucose and breakdown of glycogen into the glucose. The glycogenesis is the pathway in which glucose convert into glycogen, and glycogenolysis is the pathway by which glycogen break down into glucose 6-phosphate or the glucose. The glucose is phosphorylated to glucose 6-phosphate, and this reaction is catalyzed by the hexokinase in muscles and glucokinase in liver. As we are discussing the pathway of glycogen biosynthesis and breakdown in liver, so hexokinase is not mentioned here. Glucose 6-phosphate formed by the phosphorylation of glucose through glucokinase or hexokinase in the liver is isomerized into phosphoglucomutase into glucose 1-phosphate. The enzyme phosphoglucomutase is itself phosphorylated, and the phosphate group takes part in reversible reaction. This glucose 6-phosphate gets this phosphate and becomes the glucose 1,6-bisphosphate, and again phosphorylated enzyme is regenerated and glucose 1-phosphate is formed. During the conversion of glucose 6-phosphate into glucose 1-phosphate, this glucose 1,6-bisphosphate is formed as intermediate. Now, this glucose 1-phosphate react with the uridine triphosphate or UTP to form active nucleotide uridine diphosphate glucose or UDP glucose. This reaction is catalyzed by UDP glucose pyrophosphorylase. I am again repeating, glucose 1-phosphate and uridine triphosphate react, and the enzyme catalyzing reaction is UDP glucose pyrophosphorylase, and the products are inorganic pyrophosphate and UDP glucose. This inorganic pyrophosphate is hydrolyzed by the inorganic pyrophosphatase into 2 inorganic phosphate. As this one of these 2 product is removed, so this reaction will move in this direction. This UDP glucose pyrophosphorylase enzyme has low Km for this glucose 1 phosphate and is present in relatively larger amount, so it is not a regulatory step in glycogen biosynthesis. The initial step in glycogen synthesis involve the protein glycogenin, a 37 kilodalton protein that is glucosylated on specific tyrosine residue by this UDP glucose. De novo glycogen synthesis is initiated by the attachment of first glucose molecule to this glycogenin. Then this glycogenin molecule itself catalyzes the transfer of further 7 glucose residues from UDP glucose in 1,4-glycosidic linkage to form glycogen primer. That is substrate for glycogen synthase. First of all, this will get one glucose molecule, and then this glycogenin itself catalyzes the transfer of further 7 glucose residues from this UDP glucose, and this glycogen primer is formed. And this glycogen primer is a substrate for this glycogen synthase enzyme. This glycogenin remains at the core of glycogen granule. Now this glycogen synthase catalyzes the formation of glycoside bond or the glycosidic bond between the carbon 1 of glucose of UDP glucose and carbon 4 of terminal glucose residue of glycogen or the glycogen primer, liberating this UDP. The addition of glucose residues to pre-existing glycogen chain or primer occur at the non-reducing end, that is the outer end of the molecule, so that the branches of the glycogen molecules become elongated as successive alpha 1,4 linkages are formed. Now, the branching involves the detachment of existing glycogen chain. When a growing chain is at least, we can see better this branching in the next slide. When a growing chain is at least 11 glucose residues, branching enzyme transfer a part of it, which is at least 6 glucose residues, to the neighboring chain to form a alpha 1,6 glucosidic linkage and establishing a branch point. You can see this is the alpha 1,4 glucosidic linkage or the glucosidic bond found in here and here, and this is the alpha 1,6 glucosidic bond. Here it is located, alpha 1,6 glucosidic bond. These are the labeled glucose molecule just for the understanding, and these are the unlabeled glucose residues. Initially, as we have already discussed, the glycogen synthase act on the glycogen primer or the glycogen molecule, and the growing of the chains takes place. When this chains at least have 11 glucose residues, we can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Then a portion of it, at least 6 glucose residues, involves detachment of existing glycogen chains. We can see the alpha 1,4 glucosidic bond. Here it is alpha 1,4 glucosidic bond, and this is the unlabeled glucose residues. These are the unlabeled glucose molecules, and here it represents the alpha 1,6 glucosidic bond. Here it is alpha 1,6 glucosidic bond. Here it is alpha 1,6 glucosidic bond. Here is alpha 1,6 glucosidic bonds, and these are the 1,4 glucosidic bonds. We have discussed how this glycogen synthase forms the straight chain. When a growing chain is at least 11 glucose residues long, we can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. The branching enzyme transfer a part of alpha 1,4 chain, at least 6 glucose residues, to a neighboring chain to form alpha 1,6 glucosidic linkage, and by this way they establish a branch point. Now these branches grow by further addition of glucose molecules linked through alpha 1,4 glucosidic bond, and then further branching may also occur. And how this glycogen molecules having 1,4 glucosidic and 1,6 glucosidic linkage at branch point is formed. This is how the glycogen molecules having 1,4 glucosidic chain, straight chain, and 1,6 glucosidic. This is how glycogen molecules having 1,4 and 1,6 glucosidic linkage is formed.